From the editors of Biological Psychiatry and the Society of Biological Psychiatry, this is Biological Psychiatry Live, and I'm Dr. Tamar Gur, the social media editor, and I have with me today Dr. Seney and Dr. Ryan Logan. Could you please introduce yourself?
Hi, I'm Seney, and I'm an assistant professor in the Translational Neuroscience program and the Department of Psychiatry at the University of Pittsburgh.
Hi, I'm Ryan Logan, associate professor in Department of Pharmacology and Experimental Therapeutics at Boston University Medical Center.
Thank you both so much for joining me today to discuss your recent publication in our journal. If we could start, what was previously known about sex differences in rhythms of known circadian genes?
Sure. So there was a little bit known. There were just a couple of studies that had been done previously, really concentrating on, like you said, known circadian genes that had been, we would have predicted to be rhythmic even in the human brain. And it was found that males and females both had rhythms in these genes, but that these genes seemed to peak just a little bit earlier in females compared to males. So that was the only bit that was known previously.
Really not a lot. And
no.
How did your methodology in this study address these gaps in our knowledge?
Yeah, so we've been further developing what's called this time of death analysis, where we use basically the time of death of each subject to kind of recreate time points across a diurnal scale. And then we can look at the changes in the changes in expression of various genes across that diurnal time scale. And we were really stemming our work from previous publications from Nico Samrikian's group at McGill and Huda Akil and William Bunny's group. And one of the things that we wanted to do, based on kind of the limited knowledge that we had in what circadian gene expression looked like in males and females, and also in the human brain, we really wanted to create a well thought out, solid cohort of subjects where we could isolate these sex specific effects in a transcriptome-wide approach. So we used RNA sequencing with our time of death analysis in a well thought out cohort to look at the contribution of male and female to sex specific patterns in transcriptional rhythms in both the dorsolateral prefrontal cortex and the anterior cingulate cortex.
And it's my understanding. Oh, go ahead.
Oh, good. I wanted to piggyback off of what Ryan said. Just previously, we were never really able to do this sort of sex-specific analysis because the previous cohorts just didn't have enough females to be able to ask that question.
That was going to be my follow-up question. Perfect.
Great minds think alike. Yeah.
So what new information does your study provide mechanistically?
Sure. So one of the things is we replicated previous studies showing that both males and females have rhythms in these known circadian genes. But as Ryan said, when we expanded to the whole transcriptome, that's really what we were going after. What happens then? And what we found is that there really does seem to be some sex-specific rhythmic genes in the Lpfc and anterior cingulate cortex in males and females. And one of the really surprising things was the high number of rhythmic genes in the female anterior cingulate cortex. And then these were about 4 times as many rhythmic genes as compared to males. And these are really distinct, only rhythmic in females. And surprisingly, these genes really, across many different levels of analyses, really loaded onto synapse-related genes, which was surprising and really interesting to us. And also suggests that there might be some sort of upstream, potentially circadian related transcription factor that might drive some of this synapse related genes and being rhythmic in specifically females and not in males. And the other thing is we found this discordance of rhythmic genes between the anterior cingulate cortex and DLPFC, specifically in females. So what this is we can say, okay, can we identify the genes that are rhythmic in one brain region and another, and then ask, do they peak at similar times across those brain regions? And if they do peak at around similar times, that's considered to be concordant. And we do this not just for a single gene, but for, transcriptome wide. So transcriptome wide, are we seeing concordance in the phase of rhythms across regions? And then so we asked, is this true, and is it true in both males and females? And we found that this concordance is a true phenomenon in males, but not in females. So in females, we found this discordance in between phase in the DLPFC and anterior cingulate cortex, which was really exciting to us.
Mm-hmm. And how could these findings, for example, the discordance between in the females, how could that potentially contribute to their vulnerability to psychiatric disorders? What are your thoughts?
Yeah, I mean, the simple answer is we don't really know. I think it's a major subject of interest and active investigation in Marianne's lab and in my lab through some collaborative projects. I think, our current thoughts are that there is definitely something functionally relevant when you have concordant phase of circadian gene expression between two brain regions, especially two brain regions that are known to form circuits and regulate, behaviors that we typically care about in the context of psychiatric disorders, such as mood-related behavior, substance use-related behavior, and so forth. And I think one of the things that Mary and I are really interested in doing now, and actually have an active area of investigation in a similar context, is can we disrupt rhythms between nodes of a given circuit to see what the consequences are on behavior. And we're using some of our newer mouse models and some of our newer gene targeting tools to do that. So it's definitely an area of interest. We just don't know what it means in the context of sex, and we do not know what it means in the context of pathology.
Mm-hmm. Well, I'm excited to see you guys toggle back and forth, and I'm excited to see what comes out of this exciting collaboration. So thank you both so much for taking the time to speak to me today.